Hello everyone. I'm going to tell you a little bit about the work of the ENIGMA consortium, which is a very large network of scientists worldwide from 35 countries studying factors that help or harm the brain using medical imaging and genomic analysis. So we all know a little bit about brain imaging. It's possible to collect images of brain structure and function from large numbers of people, either people who are healthy, or young children, or elderly people, or people with a variety of different neurological and psychiatric conditions. And another interesting thing is to try and understand how the changes in the brain in these different disorders link with pathology at a cellular level, or gene expression, or even variants in our DNA genetic code. So there's been a great increase in the amount of biological data. Biobanks are emerging where large numbers of brain scans and blood test results are stored along with other associated clinical data. And the ENIGMA consortium was really founded about 6 years ago to start the largest study of the brain in the world using imaging and genetics. Its goal is to discover factors that help or harm the brain. We analyze brain scans and genomic data from currently over 33,000 people. And this data was collected in 35 countries of the world, 185 institutions. The project's really a large team science effort that involves over 500 scientists. And they're all working in about 30 different working groups that have different goals and different interests. And some high-impact papers in Nature Genetics and Nature have come of this collaboration. So there are 12 brain disorders that ENIGMA studies. We're most of all interested in how they harm the brain and what the main modulators are, such as medication, treatment, other risk factors. Within psychiatry, we study schizophrenia, major depression, bipolar illness, post-traumatic stress disorder, and obsessive compulsive disorder. There are a number of developmental disorders in children that we study. There's 22q deletion syndrome, autism spectrum disorder, and attention deficit hyperactivity disorder. We're also interested in knowing the effects of addictive drugs, alcohol, for example, on the brain. We have projects studying brain scans worldwide from people with HIV/AIDS to try and understand the effect of this infectious disease on the brain. And also there are working groups just beginning in ENIGMA on epilepsy and Parkinson's disease to try and understand what treatments are best and which brain differences most consistently found in databases worldwide. So here's a map of the people who are taking part in ENIGMA. It is the largest ever worldwide analysis of brain scans and genomic data. As I mentioned, there are 35 countries involved. The colors in this picture show you the particular diseases that the group specialize in. There are centers worldwide contributing data from autism, addiction, patients with HIV and post-traumatic stress. Other centers contributing data from people with major depression, OCD, bipolar illness. And obviously, by trying to understand the patterns in this data, it might be able to develop a fuller picture of what these diseases do to the brain and how the best treatments resist the brain changes. So one of the things that such a large effort has made possible is genetic analysis. And so we do have enough data now to do genomics with brain scans. In other words, you can screen all the millions of letters in our DNA to try and understand. So ENIGMA is also named after the ENIGMA machine. So in the Second World War, there was a major effort to decipher coded messages. And ENIGMA also uses similar types of computational techniques to try and understand what is the principle of the genetic code, how does the genetic variation affect measures that you can see in brain scans, which of course are computational images. And a great deal of the work is involved in developing algorithms for trying to predict patterns in the brain detected by imaging from markers in the genome and develop sort of generative models or generative grammars to understand why these patterns should be there. So one way to find genes that are implicated in a disorder is just to compare the DNA of patients and healthy people and look in the genome for signs of difference. Now, you can also do this in imaging databases. You can look at the signal in an image. It might be a measure of brain function, or it could be the size of part of the brain or the strength of a connection, and see if there's anything in a population that varies in the image that's linked to markers along the genome. And one reason that this is done in ENIGMA is to try and understand not just how genes affect disease, but also what it is in the brain that they're affecting, to try and understand what the basis for the symptoms might be. So one way to discover locations in the genome where common variation affects a trait such as the size of the brain or brain function or disease is to do a statistical regression at each common variant in the genetic code, look in a large group of people if there are differences in the genetic code that predict or are associated with a brain measure, and when there are, you can flag that gene as one that is associated. So in ENIGMA, what we do is the members compute brain measures from their brain scans, and we have harmonized protocols to do image analysis and quality control, and these are being tested and run at over 185 institutions. Some of the measures are the size of different parts of the brain, computed with widely used software packages such as FreeSurfer and FSL. Others, for diffusion images, look at the microstructure of the white matter, the circuitry, and there are recipes on the ENIGMA site for how to compute those if you have scans. And then in genetics projects, we go on and test associations between the brain measures and over 1 million different markers in the genome. There are harmonized protocols for imputation, in other words, converting genetic information that's been collected on one genotyping chip to a standard. And then in the final step, we meta-analyze or combine the evidence across large numbers of data sets worldwide to try and discover either markers in the genome that affect the brain or also other factors that behaviorally or in the treatment of the patient that might affect patient brain measures and outcomes. So one of the largest genetic studies of the brain conducted by ENIGMA in partnership with the CHARGE Consortium asked the question, are there any locations in our DNA where commonly there are variations that affect the size of different brain regions? And so this study revealed 6 locations in the genome that affect the size of the hippocampus, the main system of the brain involved in learning and memory. These are areas of the genome where each of us can have a different letter, and carrying the variants tends to be equivalent to about 3 years of brain aging in terms of the effect that they have on the volume of the brain. Also, we, with CHARGE in ENIGMA, have discovered common genetic variants that affect the overall size of the brain. So there's a measure called intracranial volume. It's the size of the cranial vault. It's very easy to measure. And in well over 26,000 people, there have been a number of loci in the genome that seem to affect the size of the brain. Also, encouragingly, there are tests that can be done that show that these common variants that we've genotyped account for over 1/3 of the variation in this overall brain size measure. So that's very encouraging for ENIGMA in trying to understand which they might be. Also in ENIGMA, there was a very large-scale effort published in the journal Nature in 2015 that asked the question, what are the main drivers in the genome where people have differences that might affect the size of different parts of the brain? So this studied the genetic information and brain scans of 31,000 people. And this is really just a picture of the parts of the brain subcortically and the genetic association plots, they're called Manhattan plots, indicating markers that common variation at those loci was discovered to affect the size of these different regions of the brain. Now, some of the genes that affect the size of parts of the brain are in genes that guide brain cells to the right place, one called kinectin. Some are oncogenes, genes involved in cancer that affect the proliferation of brain cells. Other genes that affect the size of parts of the brain are involved in cell death. There's one rather poetically called harakiri. It's a suicide gene that causes cells to die. Now, once you've found the genes, you can look in more detail on the genome, which are the markers that seem to drive the effects. You can look at the effects of these genetic loci on the expression of genes, plot back into the images areas that seem to have the greatest variation in relation to the genes. There's a project called ENIGMA Shape that does this. So all beginning to link genetic variation with brain variation in a very interesting way. So also there is parallel work, our colleagues in Memphis in Rob Williams' lab have been looking at homologs of these genes in mice. And they've been studying outbred mice for some time, and there are even homologs of the human genes that affect the size of brain regions that seem to affect the size of similar brain regions. Now, one interesting question is whether common variants in the genome have a bigger effect on the brain than they do on risk for disease, meaning is the percent of the variation in brain structure explained by any of these genes any bigger than the percent of variation in risk for brain diseases. And so far the data is mixed. This is a project with a psychiatric genomics consortium. In the next slide you can see in a little more detail the major SNPs or common variants that appear to affect the size of parts of the brain are plotted here. Each one accounts for less than 1% of the variation in the population, but in aggregate it can add up to rather a lot. In fact, almost about a third of the variation can be explained by common variants. You'll also see the parts of the genome that have the strongest associations with height. These have been replicated very consistently, and those that are involved in risk for schizophrenia. So it's early days yet, but it's interesting to try and understand whether the genetic variants affect the brain more in a sense than they affect risk for the associated illnesses. ENIGMA also has very large-scale studies of 12 brain disorders. These are the people who lead the efforts in schizophrenia, bipolar illness, major depression, ADHD, and addiction. And these efforts to understand brain disease use brain scans collected around the world. There are around 30 working groups with multiple sub-projects looking at different questions. There are also groups focusing on obsessive-compulsive disorder, the effect of HIV infection on the brain, 22q deletion syndrome, autism spectrum disorder, and also a group that looks at brain changes over the human lifespan. So these working groups have produced recently these two papers in Molecular Psychiatry, the largest ever surveys of brain scans to try and understand what's different in schizophrenia or what's different in major depression. And really it's just a worldwide effort, very exciting, that's honing in on certain parts of the brain that are most strongly implicated in these diseases. So in schizophrenia, this working group, led by Theo Van Oop and Jess Turner, found that the hippocampus, the part of the brain involved in learning and memory, is smaller in people with schizophrenia, at least on average. The basal ganglia tend to be enlarged, at least on average, especially in people that are taking second-generation antipsychotics. And then the ventricles, the fluid-filled spaces in the brain, tend to be much larger in schizophrenia patients than healthy people. In bipolar disorder, different projects being led by Derek Heibar and Ola Andreasen, a large number of people who are expert in bipolar disorder around the world coming together in this project. The brain differences are somewhat smaller than those found in schizophrenia, although the hippocampus is also smaller in bipolarness than healthy folks. And again, the ventricular system, the fluid-filled space in the brain, is enlarged. Now you can start to compare the different psychiatric illnesses. So here we've plotted the amount of brain difference there is for a range of different structures in schizophrenia, bipolar illness, and major depression. And one of the clearest messages is that the brain differences appear to be greatest in schizophrenia, somewhat intermediate in bipolar illness, and somewhat milder in major depression. But there are some parts of the brain, the hippocampus, for example, which is involved in learning and memory, that are altered in all three of these disorders, making it interesting to see if there's a signature of different disorders in the brain. In depression, it looks like the hippocampus, the learning and memory system, is on average smaller, particularly so in patients who've been ill a long time. Chronically ill patients have much greater differences, with very little differences at the first episode of depression. Much smaller hippocampus if the person is diagnosed before age 21. And again, these are the general statements that apply on average, not to everybody. There also seems to be hypertrophy of the caudate in groups of people with major depression who are taking antipsychotics, and not all of them are treated with those medicines, but ENIGMA is beginning to reveal the pattern of the medicines on the brain. Now, one of the most exciting directions in ENIGMA is to map patterns of brain differences across different brain diseases. So if you look at this slide, there are different parts of the brain and how much different they are in size on MRI between people with schizophrenia and healthy people with bipolar illness and healthy people, and similar things in depression and ADHD. You'll begin to see that there's sort of a profile of differences that appears to be unique to each illness. Some of the deficits overlap, and these are really beginning to hone in on what are the important differences that characterize or typify these brain disorders in 33 countries of the world. Now, we talked about also not just comparing patients and controls, but also looking in the genome for spelling differences or genetic variants that affect the size of parts of the brain. One thing that you can do, and this is active work in ENIGMA, is go through images pixel by pixel and say, does any of the signal in this part of the image relate to genetic variation? There's something called a voxel-wise genome-wide association study. It's very, very computer-intensive. You're doing over 1 trillion tests. This can generate a huge amount of data, 200 terabytes of data on associations between genetic variation and images, but certainly a very exciting one for cracking the enigma of how brain variation relates to genetic variation. Now, one of the ways that we're doing this in ENIGMA, searching images and searching the genome for connections, is essentially one by one going through image databases, aligning them to common coordinate systems, common brain templates, and all of the data that's produced at each site doesn't need to be transferred to the other sites. We're coming up with this is an approach developed by Nada Jahanshad and others in ENIGMA, essentially to do genomics images at a large range of different sites, essentially by measuring brain volume differences using a computer program, plotting them into the image, and then at each site trying to understand what are the variants in the genome that predict these, and then aggregating the evidence across sites to create an aggregate picture of what is going on. Another way to think about this is as a multi-party private distributed computation. If there are a large number of sites that have images and genetic data, you can fit a machine learning model, essentially find predicts in the genome that explain some of the variations in the images. You can use sparse learning methods or compressive sensing methods and others, essentially to find an economical summary of the genetic markers that are predictive of variations in the brain scans. And these can be done using massively parallel computations, so that's another area that we're currently pursuing. One of the exciting applications of ENIGMA is in developing norms for brain development and also for aging. So we're all familiar with maybe going with a child to a pediatrician and seeing if their height and weight is consistent with the normative data available from other children. And one of the things that we are doing in ENIGMA as part of what's called the Lifespan Project is computing the size of parts of the brain in people across the lifespan, age 2 to 92, compiling data from 33 countries, over 10,000 people, and really looking at the growth charts for different parts of the brain. This is for part of the brain, the right thalamus, and you can see that this grows until you're in your early 20s, and then now, this is useful because, obviously, in data from around the world, you can decide whether or not the brain metrics are reflective of normality, or whether they fit with our expectations for people of a particular age. So applications obviously include trying to check whether brain measures are within the normal range, if a treatment has restored them to the normal range, or if there's been significant decline in brain structure and function. So these ENIGMA lifespan curves, it's a project led by Denai Dima and Sophia Frangu, are beginning to be compiled for a large range of different structures. Some structures of the brain grow rapidly and then plateau and then shrink. Others just increase in size. The fluid-filled spaces increase in size the older you get. And so this is also an interesting way of sort of predicting a person's brain age on the basis of their brain scan. Now, also, some of this reference data is quite useful for doing epidemiological studies. Now that we know that some parts of the brain develop rapidly in early life and some take a longer time, it can begin to help interpret the timing of some of the developmental and psychiatric disorders that occur in early adulthood in relation to the structures that are developing the most at those times. You can also draw from this data some quite general statements about at what age people tend to have the greatest volume for different parts of the brain. There is a sequence. There tends to be an earlier maturation for primitive parts of the brain involved in vision and sensation, later maturation for parts of the brain involved in language function, and then even later maturation for parts of the brain involved in self-control, which is a sort of interesting observation about behavior as well to go along with that. So these are normative data, they're percentiles from the ENIGMA lifespan group, really just showing that as we age, there are very different patterns of aging or structural change for different parts of the brain that are listed here. So just to summarize, ENIGMA is a large worldwide study of the brain. We study 12 major brain disorders, but we also are able to do genetics, so that there is the power in such a large data set to see the effects of single letter spelling differences in the genome. Some of these genetic differences affect brain volume by as little as 1%, but you can still find them based on aggregating so much data. It's certainly a team science or crowdsourcing effort. Scientists from 307 countries are taking part. 307 scientists are taking part from 35 countries. And this work is organized into 30 or so working groups that answer different questions about different diseases, symptoms, and genetic risk factors. Some of this leads us into questions that use big data. Certainly, searching scan databases pixel by pixel and nucleotide by nucleotide in the genome requires trillions of tests. But some of the effects ENIGMA has found, they're just quadrillions to one in terms of statistical significance, so it's perfectly okay to do a large number of tests. Some of these are screening the connection matrices of the brain networks, and genomes at the same time. And because of the large volume of data, this is being accelerated by distributing the computation across very large numbers of sites. It has analogs in machine learning in areas such as multi-party distributed computation. Now, there's a big thank you to all of the people that have contributed to ENIGMA. Here are groups of ENIGMA members at Amsterdam, people in Italy here from a consortium called Imogen across Europe, and also members. So finally, an acknowledgement is due to all of the people that fund ENIGMA. In the United States, the NIH Big Data to Knowledge Program, the BD2K program, has funded ENIGMA. Imogen and other consortia in Europe tend to be funded by European federal and private agencies. And also the private and federal funding to all of the well over 300 ENIGMA co-authors in institutions around the world, some of them listed here. And without this support and the support of the patients and volunteers, none of this work would have achieved the success that it now has. So if you want to take part in ENIGMA, you're very welcome to. You can go to this website, look at different projects that might be of interest to you, and certainly if you have either brain scans with clinical data or brain scans and genomic data, there are large numbers of projects that you might be able to get involved in very quickly. Thank you very much.